Hello, this is Kevin Kong, and my presentation is on the 2018 paper by Inquimbert et al. Titled NMDA receptor activation underlies the loss of spinal dorsal horn neurons in the transition to persistent pain after peripheral nerve injury. Firstly, the background of this paper is that pain resulting from nerve injuries is chronic and persistent. Prior research has linked this chronic neuropathic pain to apoptosis of neurons in the spinal cord. The mechanisms for this apoptosis has been disputed, however. Glutamate is a major excitatory neurotransmitter, and GABA is a major inhibitory neurotransmitter in the spinal cord, and NMDA receptors increase signaling after injury, which can lead to excess calcium flux, causing excitotoxicity and cell death. With this, the researchers investigate whether nerve injuries produce an excitotoxicity challenge in the spinal cord. To test this question, researchers generated mice that lacked NMDA receptors and caused spare nerve injuries on the sciatic nerve. They then quantified spinal neuron apoptosis, recorded patch clamp electrophysiology, and measured the withdrawal responses to noxious stimuli. Figure 1 shows the SNI performed on the mouse sciatic nerve. Panel B shows the staining of a section of the spinal cord, where the green is a marker for apoptosis, the blue stains for nuclei, and the red indicates locations of neuronal nuclei. Panel C shows the locations of apoptosis between a sham surgery and an SNI surgery, and the SNI mouse has significantly more apoptotic markers. Continuing from Figure 1, the researchers used a GRIN1 knockout mouse created from the Krelock system. GRIN1 is a subunit of the NMDA receptor, and its deletion renders the receptor non-functional. NMDA application to these mice show no current in the patch clamp electrophysiology compared to mice that did not receive the CRE. The rightmost panel shows the main takeaway from these experiments, which is that the mice with GRIN1 deleted have significantly less apoptosis in the spinal cord after receiving a nerve injury. To confirm this, the researchers stained the dorsal horn for neuronal nuclei protein, a protein expressed exclusively in the nuclei of neurons. They found that indeed, SNI decreased the number of neurons in the dorsal horn of the spinal cord, but not in mice that had GRIN1 deleted. Next, the researchers performed patch clamp electrophysiology to measure the miniature inhibitory postsynaptic currents on mice with GRIN1 deleted, and SNI found that GABAergic MIPSCs decreased significantly in mice with intact NMDA receptors, but show no significant change in mice that had the receptor deleted. Thus, GRIN1 deletion prevented a decrease in inhibition of the dorsal horn neurons. To verify this result was indeed from the death of neurons and not from SNI interrupting the GABA synthesis or transport pathways, the researchers quantified the expression levels of GAD1 and GAD2, which are essential enzymes in GABA synthesis, and GLYT2 and VAT2, two proteins involved in the vesicular trafficking of GABA. Using chromatin immunoprecipitation for histones of active gene transcription, levels of these four proteins were quantified. Levels of all these four proteins show no significant decrease between mice that had no injury and mice that had SNI, which confirms that the decrease in GABA was indeed due to NMDA signaling and not due to downregulated GABA synthesis or transport proteins. Consequently, the researchers investigate the effects of NMDA receptor deletion on the development of chronic pain. Using SNI and GRIN1 knockout mice, they perform noxious assays using mechanical stimulation from von Frey filaments and cold stimulation from acetone evaporation. As seen in panels A and C of this figure, both mechanical and cold hypersensitivity return to the baseline withdrawal response in mice with GRIN1 knocked out. Acute neuropathic pain does not progress to chronic pain in GRIN1 knockout mice, which shows that the NMDA receptor is necessary for the development of chronic hyperalgesia. The researchers then repeated many of the previous experiments using mice that had BAX deleted constitutively. BAX is a protein that regulates cytosolic and ER calcium levels and is a key protein in intrinsic apoptosis. Like previous results, BAX deletion decreased the amount of apoptosis in the dorsal horn after SNI, and BAX deletion does not decrease miniature inhibitory postsynaptic currents. Furthermore, BAX deletion attenuated the chronic hyperalgesia seen in mice following SNI surgery. This closely mimics the results seen from the deletion of GRIN1. This study shows that deleting NMDA receptors protects against neuropathic apoptosis in the dorsal horn of the spinal cord and provides evidence that functional NMDA receptors are required for the development of chronic pain from acute pain caused by nerve damage. BAX deletion follows a similar pattern to GRIN1 deletion, so these proteins may be candidates for future pharmaceuticals in the treatment of chronic pain. Although not quite feasible in adult humans, this study shows the neuroprotective power that a gene deletion can have over hyperalgesia. Finally, GABA transmission and the total excitatory balance of the nerve may be a key target in therapies that target chronic pain as well. And this concludes my presentation.